Hello, and welcome to the JASN podcast, the monthly podcast where we highlight an exciting paper from the current issue of the journal. The papers may be clinical, basic, or translational, but they're chosen to reflect the diversity of JASN content. I am Dr. David Ellison, professor of medicine and chemical physiology and biochemistry at Oregon Health and Science University, and also a deputy editor at the journal. Today, I'm joined by Yasmine Lund-Ricard and Agnès Boutet to discuss their interesting paper entitled Postembryonic Maintenance of Nephron Progenitor Cells with Low Translational Activity in what I'll call the Catshark. They can provide the official name of the organism, and this appears in a recent issue of JASN. So we're delighted to have them with us today. And if you just please introduce yourselves and tell us a little bit about your background.
Okay, so maybe I can start, David. Thank you very much for offering us the possibility to do this podcast. It's very interesting. So I am Agnès Boutet. I am a researcher in molecular and cellular biology, and I'm working in the marine station of Roscoff, which is located in northern Brittany in France. And I am also teaching duties, and I'm in charge of a course in developmental biology for bachelor students, and also master course for master students on the use of marine organisms in experimental biology. So I have the two jobs, researcher and teacher. And so in my precedent research, I was interested in kidney development and in kidney fibrosis, but using the mouse as a model. So this paper on the Catshark was quite new for me as well.
Okay, hello. So my name is Yasmine Lund-Ricard. I'm a molecular and developmental biologist, and I'm currently working in Galway, Ireland. I studied biology in Paris at La Sorbonne, and I discovered the research station in Roscoff during my master's, and that's when I met Agnès also. And I became very interested in this topic of progenitor maintenance in cat sharks because I could think about very broad concepts like what maintains stem cells, how translation rates affect cell fate, and how the evolution of development can impact regeneration.
Great. Well, as I said, it's great to have you both here. We don't publish a lot of papers using fish as a model background or other aquatic animals to inform us about biology. Although, one of my great experiences was reading Homer Smith's book, From Fish to Philosopher, which I don't know if either of you have read it, but it's near and dear to my heart. In addition, I just should mention that my own career has really been focused for more than 35 years on the thiazide-sensitive sodium chloride cotransporter in the kidney. We focused mostly on the mammalian transporter, but it was actually first cloned from the flounder bladder. So the history of the use of aquatic organisms to tell us about the kidney in other animal species is really long and storied. But I just wonder, and I think we heard a little bit about this, but it sounds like this may be a new model system for you both. Can you tell us a little bit about, I think Yasmine told us a little bit about how you got into using the Catshark as a model system, but tell me a little bit more about your history working with fish and aquatic organisms.
Okay, so I discovered the Catshark. So the Latin name is Scyliorhinus canicula. So now you know it. So the Catshark as a model, when I got a permanent position in Roscoff, so it was more than 10 years ago. And of course, the precedent papers revealing the presence of a continuous nephrogenesis in cartilaginous fish was a starting point to investigate the nephron progenitor cell population. But I have to say that the marine station of Roscoff was created in 1872, and its founder, Henri de la Casse-Dutier, started at the same time a journal to report the work performed at the station. And in this journal, which was called the Archive of Experimental and General Zoology, in one of the first volume, we can see some beautiful drawing of Catshark embryos. So this means that it was already a matter of interest at that time, so more than 150 years ago. So of course, they were not studying the kidney, but they were interested in this shark because it's a small shark, and it was amenable for study. So it is important that we never start to say that we never start from zero. And in this case, we can rely on the legacy from the past. So so we cannot say that Catshark is an emerging model. It is really a model that was on stage for a long time. But of course, the topic of studying the kidney, the maintenance of the Nephron progenitor cells was more recent. So Yasmine, you want?
Yeah, I'd like to add something. So what's interesting about sharks also is that they're part of the cartilaginous fish family, and this family is the sister group to the vertebrate family. So the sharks and other cartilaginous fish are really interesting in studying the evolution of development and understanding what has been lost or gained during the evolution of vertebrates. And it seems like the fact that sharks can regenerate their kidney is actually an ancestral trait that was lost then later in mammals. So it's very interesting to look at kind of a diversity of animals to understand what's happening actually in humans.
Yeah, that's interesting. So that actually leads to the next question, which is sort of as a layperson, you say that the ability to generate new nephrons is lost during evolution. And I wondered, is there anything specific environmentally about cat sharks that would make it useful or helpful to develop new nephrons that maybe isn't found in other organisms where that ability has been lost? I mean, why do you think it was there and then evolutionarily it disappeared over the years?
So this is a very interesting point. The fact is, also personally, I don't know, but it is true that in mammals, so the mammals cannot do a continuous nephrogenesis, but they are able to answer in case of kidney injury. So in this case, the nephron will, well, you might know that very well, but the nephron will become hypertrophic. So if the kidney suffered damage, the nephrons will become larger, bigger, so hypertrophic, and to increase the capacity of filtration of the damaged organ. So it seems that there is not a specific advantage for shark to regenerate, so to do a continuous nephrogenesis. I would say that it seems that fish, sharks, and mammals display different strategies to respond to kidney injuries. So fish and sharks will make new nephrons, while mammals are able to increase the size of the remaining nephrons to adapt to the new condition in case of there is a kidney damage.
Yeah.
So if there is an environmental factor that is involved in this, well, clearly I don't have the answer.
That's super interesting. I'm a physician scientist, so we deal a lot with the reactive hypertrophy of nephrons that occurs after kidney damage, and while that can maintain the glomerular filtration rate initially, we now think that actually contributes through hyperfiltration injury to long-term kidney damage. So in some ways, if you're able to generate new nephrons, you don't have to hypertrophy your kidney, and you might avoid kidney damage. But let me get a little bit more into the results of the paper. So to identify the nephron progenitor cells, you use BRDU pulse chase labeling with a long chase time. So I wondered if you could just provide a brief overview of this technique, how it works, and what the long chase time tells you that shorter chase times don't.
Okay, so we wanted to locate these progenitor cells in the cat shark. And so we know that in humans and other animals, adult progenitor cells have a slow cycling population. To find the slow cycling population, we were using BRDU. And BRDU will stain cells that are going through S phase, so that are proliferating. This is because BRDU is a thymidine analog, so it will incorporate into newly synthesized DNA. And so we proceed with injecting BRDU into juvenile sharks, and then we look at the BRDU signal over long periods of time. So close to the injection, we can see which cells are proliferating. So we see that the nephron epithelial cells are highly proliferative. But over longer periods of times, we can see that cells have retained this label and are thus slow cycling. And so with this approach, we were able to describe a population of nephron progenitor cells that are slow cycling.
And so if this same approach is used in a mouse model, for example, do you not see those slow cycling cells?
No.
But if I remember well, they found slow cycling cells in the renal papilla in the adult mouse.
Okay.
And because of course, well, this is different type of cells. This is not the nephron progenitor cells, but they have identified already some cells that they called stem cells in mammals, and it was in the renal papilla. But it is a different lineage from the 6-2 positive cells that we see in the embryo.
Yeah. So a key finding, at least from my reading of the paper, is that protein synthesis is higher in epithelial cells than in progenitor cells, or maybe better to phrase it that the protein synthesis is lower in the progenitor cells, which kind of makes sense to me. But do you see this as a key component of the signal to retain the progenitor phenotype? I mean, why is it that they're cycling more slowly like this?
The next step that we would like to investigate is what is the signal that impair the Catshark progenitor cells to be exhausted as it is in mouse. So in mouse, we know that during embryogenesis, the nephron progenitor cells will have to choose between self-renewal and differentiation. And around birth, so they are all definitively, they lose definitively the capacity to self-renew, and they will all go for differentiation. And so previous studies have shown that the Wnt pathway has an important role in controlling this balance between self-renewing and differentiation. And to link this data with translation regulation, because we found indeed that when the cell differentiate, they increase translation and global protein synthesis. So to link this part with the Wnt pathway, we can hypothesize that the specific inhibitor of the synthesis of Wnt protein should be involved in the maintenance of the progenitors beyond birth in Catshark. So this is a hypothesis. And I think that we need to go for a global analysis of the nephron progenitor cell proliferation before hatching in Catshark and after hatching in Catshark to see what is changing between at this transition step, actually. So we have ideas about the molecule that is involved, but we need to make the experiment to validate the hypothesis.
Maybe that will be your next paper.
I hope.
So the other thing that I wondered about, again, and I'm not a developmental biologist, so this may be naive questions, but it seems as if what you observed is this slowly cycling population of progenitor cells sort of beyond early development. And yet, as development occurs, it seems to me one has to have the progenitor cells cycling rapidly if you're going to make a new kidney. Do you think that there's a change in the cycle length to make it so that it starts out rapid so you can generate or develop your kidney, and then later on slows down so you have a progenitor subpopulation that's available for other things?
Well, the thing is that I think that there are different populations with different speed, cycling speed in the nephron progenitor cells. And we can assimilate the cells that have a longer cell cycle length to cells that display a dormant or quiescent state. So they are able to self-renew and at a very slow rate, and to assure continuous production of the stem cell within the niche. And in mice too, they have identified also two populations of progenitors. that display different cell cycle speed. And this actually correlates with, if I remember well, with the level of 6-2 expression. So it seems that the cells that have a high expression of 6-2 have a long cell cycle, while the cells with low 6-2 expression display a faster cycle length, and actually they are located closer to the site of nephron formation. So they have also two locations. So it seems that, so actually the population of stem cells is not as homogeneous as we thought. And we can analyze this with the single cell RNA-seq. Now we can indeed reveal that in a stem cell population, you have many subpopulations. And indeed, what we observed, but it was also observed in the mouse, it's you can have different cell cycle speed in this stem cell population.
So in many ways, I think the most exciting and newest part of the observations in the paper revolve around mTOR. And so mTOR, as I understand it, is higher in the epithelial cells than in the progenitor cells. And as mTOR or the various types of mTOR are areas that can be manipulated pharmacologically, I think that raises the possibility of intervention or change. So do you see the possibility of sort of reintroducing nephrogenesis in mammals by manipulating mTOR? That would obviously be an exciting avenue to explore, since many of us clinical nephrologists see a lot of acute kidney injury developing into chronic kidney disease, and seems like that may be a holy grail. So I wonder if you can comment on the role of mTOR and how you see this moving towards translational potential in the future.
Yeah, so unfortunately, the things are not so simple. So of course, so manipulating mTOR in the mouse have been done previously in the lab of Raphaël Copon in the States. So they did a conditional knockout of mTOR in the 6-2 positive cells. And this revealed that what they observe is that there are several tubular lesions, meaning that the absence of mTOR during embryogenesis is above all deleterious. And it's quite normal because mTOR is a very important pathway. It is involved not only in the initiation of translation, but it is also controlling a lot of cellular processes. So I don't think it's so it's quite normal that if you delete, even if in a specific cell type, if you delete mTOR, well, you will affect many other aspects of the cells, and this is why it is deleterious. And so yes, so in this paper, they show that it's deleterious, and so, but they found that the conditional deletion of TSC1, which is an inhibitor of mTOR, could induce an increase of nephron endowments, if I remember well. But this was mTOR independent. So it seems it's quite complex. But the fact that mTOR, so we identified indeed that the mTOR is activated when the progenitor cells are differentiating. This is consistent with the previous study that were made in the mouse, because it's the lab of Raphaël Copon, who did also transcriptomic analysis of the nephron cell population, nephron progenitor population at different time points of embryogenesis and at birth. And what they found is that as the cells are exhausted, they increase the mTOR mRNA. So and you find a lot of activation of proteins in relation with ribosome synthesis and biogenesis and other process involved in translation. So it seems that, and it's quite normal that it's a general process that when the cell differentiate, they increase the translation. But to find the key element that is permitting the maintenance of the progenitor cells all along the life of the cat shark. We need a broad approach, a high throughput approach.
Is that something you all are planning to do? Are you planning to take that high throughput approach and find these keys? Yeah.
Yeah, we hope to have it. Yeah.
Great. Well, that's the last of the questions that I have. I don't know if either of you want to add any last comments about the work or where it's going, or whether Yas, where you're going in your future in terms of your own work.
Yeah, I'm actually working now with a model that's a cnidarian, so quite far from the cat shark now. So I'm working on Hydroctinia, but I'm kind of continuing this thought experiment of wondering how stem cells are maintained after development, because Hydroctinia maintains pluripotent stem cells after embryonic development. It's a very unique case. And so I'm starting a project on that, trying to understand what are the mechanisms behind this maintenance. And I am interested in mTOR still. I think it's a pathway that will definitely be involved in the maintenance. One last thing I can add is that so mTOR activity works in the initiation of protein synthesis, but also low levels of mTOR activity, you have autophagy, the process of autophagy happening. So this is a catabolic protein degradation process. And we know that this process is actually necessary for stem cells to both in quiescence and in self-renewal. So I'm really excited to see how I will connect these things together and maybe even connect them to the cat shark to kind of understand a bit more about the evolution of the processes that control stemness.
Yeah, I just want to add that it's good to see that Yasmine kept the general interest for stem cell, for the study of stem cell. So now she's a postdoc. She was a PhD student, and she can move from a model to another without any fears. And so it's really good to see this this progression. Sorry. So for my part, yeah, we'll start to, of course, to go more deeply in the understanding of this maintenance, and we will mainly have a high throughput approach. We also want to induce chemical injury in the kidney of cat shark to really analyze at the molecular level the mechanism of nephron formation, because this has been observed but never documented molecularly. So this will be the next step. And so it was very great to do this discussion. Really, it is. Thank you very much for giving us the opportunity to talk about our work, and in this formal small interview. It's very, really great.
Yeah, thank you so much.
I really appreciate it as well. And I'm not kidding at all. I just think that we've learned so much from fish and aquatic organisms and other earlier organisms about mammalian biology, and that's really been key to so many of the advances. It also reminds me that there is a program in the US called Rebuilding a Kidney, and in a sense, that the holy grail goal is to be able to rebuild kidneys so we don't have to use more cumbersome approaches to keeping people's kidneys healthy, and learning more about how we can enhance or increase the number of progenitor cells, and the kidney can rebuild itself would certainly be very helpful. And I really believe that we'll learn a lot from studies such as this. So really appreciate your sharing your work, and I think it's great that it appears in JASN, and it keeps this idea alive that those who are working in clinical nephrology need to keep learning from those of you who are working with other model organisms, and that's great to hear. So I really appreciate the time of this conversation, and I look forward to hearing more from both of you in the future about your various projects.
Okay, thank you very much.
Thank you so much.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.